Dear students, welcome to 12th class Biobotany. So in this chapter, asexual and sexual reproduction in plants. In this chapter, we already covered pollination. Okay, pollination means transfer of pollen grains from anther to stigma of a flower is called pollination. And we discussed about different types of pollination, self-pollination, cross-pollination, and what are the different types of pollinating agents, etc. Now we are going to study fertilization. Fertilization. So after pollination, pollen grains they land on the stigma of a flower. Then next is going to happen fertilization. What is fertilization? Fertilization means fusion of male gamete and female gamete. Fusion of male gamete and female gamete is called fertilization. Okay. Fusion of male gamete and female gamete is called fertilization. In case of plants, angiosperms, double fertilization present. In angiosperm, fertilization is double fertilization. So what is meant by double fertilization? Double fertilization, which means from the pollen grain, two male gametes are produced, and these two male gametes they enter into ovule through pollen tube, and inside the ovule here one male gamete fuse with egg, another male gamete that fuse with polar nuclei, and they form endosperm. So that is called double fertilization. Two male gametes they fuse, one male gamete fuse with egg, another male gamete fuse with the polar nuclei in the embryo sac. So that's why in angiosperm fertilization is called double fertilization. Okay. Then let's study what are the events of fertilization, or what are the stages of double fertilization. So events of fertilization or events of double fertilization. So what are those? The first step, germination of pollen to form pollen tube on the stigma. Second stage, growth of pollen tube in the style. And third stage, direction of pollen tube towards the micropyle of the ovule. And fourth one, entry of pollen tube into the embryo sac. And fifth stage, that is discharge of male gametes and syngamy and triple fusion. Okay, so first stage, germination of pollen to form pollen tube on stigma. So after pollination, these pollen grains they are landed on this stigma of the flower, and these pollen grains they germinate and they form pollen tube. Okay, that is the first stage. The pollen grains they land on the stigma and they germinate and form pollen tube. Okay, then next stage, this pollen tube, the growth of pollen tube in the style. This is stigma of the flower. This is style. This elongated portion, that is style. Then this is ovary. Inside the ovary what present? Ovule present. Okay, so this pollen tube which grow in the style and which passes through style, then direction of pollen tube towards the micropyle of the ovule. Okay, and this pollen tube which enter into this, which directed to micropyle end of the ovule. Okay, which crosses ovary, and inside the ovary ovule is formed, and which directed to micropyle end of the ovule. Then entry of pollen tube into this embryo sac, which enter into this ovule and finally enter into this embryo sac. So this is ovule and this is embryo sac of ovule. Embryo sac. Okay, so finally the pollen tube which is in the embryo sac. So what happens next? The pollen tube which contain male gametes. So they discharge male gametes into this embryo sac. Okay, that is a discharge of male gametes into this embryo sac. And what happened? Syngamy and triple fusion. What is syngamy? Syngamy means one of the male gamete which fuse with egg cell. Okay, so here in this embryo sac, what present? Egg cell present. Okay, that is female gamete. Egg is present. One of the male gamete coming through pollen tube which fuse with egg cell. That process is called syngamy. What is syngamy? One of the male gamete or sperm fuse with egg. That is called syngamy. And what is triple fusion? Another male gamete which fuse with polar nuclei. Polar nuclei. That process is called triple fusion, and they form endosperm. One of the male gamete, they fuse with the polar nuclei in the embryo sac and they form endosperm, that is called triple fusion. And another male gamete, they fuse with egg and that process is called syngamy. So these are the events of fertilization. Okay, so this is double fertilization which is present in angiosperms. And this all process, this all events is called pollen pistil interaction. Pollen pistil interaction. Okay, so first of all, the pollen grain which will land on the stigma and they germinate pollen tube, and next stage growth of this pollen tube through style, and next stage this pollen tube which directed to micropyle end of the ovule and which enter into this ovule, and finally in the embryo sac, and they discharge the male gametes into the embryo sac, then syngamy and triple fusion. Okay, so this all process called pollen pistil interaction. Then we will discuss in very detail what is happening here. If one pollen grain land on the stigma, how this pollen grain they are recognized by stigma. If it is pollen grains from another plant, what is going to happen? If it is the pollen grain from this same species, how they recognize, how pollen tube germinate on the stigma, and all this process we will discuss in very detail. Next, pollen on the stigma. Okay, so this is the stigma of the flower, this is style and this is stigma and this is ovary. So this all structure represent pistil. Suppose if the pollen, if it is from other plant, okay, the pollen grains from other species, they are not going to germinate on this stigma, and this pollen is from same species, then only the pollen is going to germinate. So how this compatibility of the pollen grains and germination of pollen grains on the stigma facilitated? Is there any chemicals or mechanism behind this germination of pollen on the stigma? Okay, that's we are going to study. So here the pollen grain and this is stigma, they land, these pollen grains they land on the stigma, and if it is receptive stigma of the pollen grain or the right stigma of the pollen grain, this pollen grain will germinate. And how do they germinate? That germination of pollen tube facilitated by germination of pollen tube facilitated by these two factors. One is stigmatic fluid which is present on the wet stigma, and pellicle which is present on the dry stigma. Stigmatic fluid on wet stigma and pellicle on the dry stigma, which lead to germination of pollen, germination of pollen. Okay. So these two factors, stigmatic fluid, if one pollen grain they land on the stigma, if it is the receptive stigma of this pollen grain or it is the right stigma of the pollen grain, the stigmatic fluid present on the stigma which help to germinate the pollen tube, or if it is dry stigma, the pellicle present on the surface of stigma which help to germination of pollen tube. Okay, and these two factors they also decide compatibility, and incompatibility of germination of pollen. Compatibility and incompatibility of germination of pollen. Compatibility, which means the pollen grain, which these factors, if they are present on the stigma, they allow the germination of pollen grain. Incompatibility, these two factors, stigmatic fluid and pellicle, if pollen grains are present on the stigma, they do not allow the germination of pollen grain, they reject the pollen grain. Okay, so these two factors they decide compatibility and incompatibility of germination of pollen grain through a mechanism that is called rejection, what is that, recognition or rejection protein reaction. Recognition rejection protein reaction. By this mechanism, pollen grains they allow to germinate or they reject the pollen grains. Stigma allow the pollen grain to germinate or they reject the pollen grains. At what condition they reject the pollen grain? If this pollen grain is from other plants, other species, okay, it is not possible to germinate the pollen grain on this stigma. If this pollen grain is same plant or from same species, then only this stigma allow them to germinate. So what factors decide the germination of pollen tube? The stigmatic fluid and pellicles. They allow the germination of pollen grain on the stigma. Okay, so this all mechanism, germination of pollen tube and rejection of germination of pollen grain and rejection of pollen grain by the stigma is called recognition rejection protein reaction. Okay, that decide compatibility and incompatibility of pollen grain. Sexual incompatibility also present in case of pollen germination. Sexual incompatibility. So what it means? Sexual incompatibility. Sexual incompatibility, which means the pollen grain is from another species and stigma or flower is from another species, so that pollen grain is not going to germinate. That is called sexual incompatibility, or it is also called interspecific incompatibility. So the pollen grain is from other species and stigma of flower is another species. Okay, so pollen grain is not going to germinate. And another incompatibility, that is self-incompatibility. Self-incompatibility, which is intraspecific compatibility. What is self-incompatibility? Suppose you take a bisexual flower. Okay, so there is a chance for self-pollination in that bisexual flower. So how we can prevent that bisexual flower? So plants, this flower, they take self-incompatibility. That means they do not allow the germination of their own pollen on the stigma present in same flower. They adopt self-incompatibility. They do not allow the germination of pollen, their own pollen on the stigma. Okay, so that is called self-incompatibility. So sexual incompatibility may be interspecific, that means different species, and maybe it is intraspecific, that is in the incompatibility in same species. This intraspecific sexual compatibility also called self incompatibility. So pollen grains, they land on the stigma. The first change you can see on the pollen grain, that is hydration. Hydration, which means loss of water. So pollen grain, they lose water, that is called hydration. Then what happens? The pollen grain, we studied the pollen grain structure. Pollen grain, they have wall and they lose their pollen wall protein also released. Their pollen wall protein also released. Okay, so what are the changes you can see when pollen grain land on stigma? First, hydration of pollen grain. The second, their pollen wall proteins are released. Then what is going to happen? They germinate pollen tube. Okay, so this is the pollen grain. They form the pollen tube, and all the contents present in this pollen grain, the generative cell and cytoplasm, everything that moves into this tip of the pollen grain. So this is the tip of the pollen grain. Tip of the pollen grain. Okay, so all content present inside the pollen which moves into this tip of the pollen grain, and this tip of the pollen grain which grows through the style. Okay, and here the remaining portion of pollen which is occupied by vacuole and which is blocked by a callus block nearby tip of the uh, pollen grain. So here callus plug is found. So remaining portion occupied by central a vacuole, and all the cytoplasm and the materials and male gametes, everything found at the tip of the pollen grain. And this tip of the pollen grain, which looks like something hemispherical structure, tip of pollen grain, or tip of pollen tube, sorry, tip of pollen tube. This is pollen tube. Tip of the pollen tube, which is look like hemispherical structure, and the all contents of pollen now present at the tip of the pollen, and which is blocked by callus plug, and only this pollen tube grows through style. So this structure is also called cap blocker. Cap blocker. Okay, so they have something like a hemispherical structure, the pollen tube, and this pollen tube grows and they pass through style, and finally they reach into ovule. So pollen tube in the style. The growth of pollen tube in the style depend upon the type of the style. So there are three types of style present: solid style or closed style, and next one hollow style or open style, and the third one semi-solid style or half closed type. Okay, half closed style. So let's study what are the different types of styles. So first of all, hollow style or open style. Hollow style or open style which present in plants monopod plants. Okay, and here the style region which is lined by a single layer of glandular canal cells. Okay, this is the style, and this style region is lined by glandular canal cells. Okay, and the pollen tube which grows through this canal. And these glandular cells, they are also called transmitting tissue, and they produce the secretions. That secretions they provide as a nourishment for the growth of pollen tube, and also they consist, that secretion consists compatibility factors. Okay, and what are the components present, the secretions that produced by glandular canal cells? that contain carbohydrate, lipids and enzymes like esterases and acid phosphatases and compatible controlling factors. Okay, so those secretions are produced by glandular canal cells present on the style. So that is hollow or open style, which is present in monocot plants. Then what is solid or closed type style? Here this type of style which present in dicot plants. Okay, there is a large central core of cells that is called transmitting tissue, and the pollen tube which passes through the intercellular spaces of this transmitting tissue. Okay, here they pass through the hollow space found in the style. Here pollen tube pass through the intercellular spaces of transmitting tissue, and these transmitting tissue they also produce the same secretions that consist of carbohydrate, lipids and enzymes which nourishes the growth of the pollen tube, and which also contain the factors which control compatibility. Okay, so this is solid type style and hollow or open style type. This is present monocot plant, this is present dicot plant, and third type is semi solid type style, or it is also called half closed type style. Okay, so these are the different types of styles present in plants. Next one, entry of pollen tube into the ovule. There are three different types of entry of pollen tube into the ovule. Those are porogamy, chalazogamy, mesogamy. So what it means? Okay, so there are three different types of entry of pollen tube into this ovule. So this is ovule. You know already the structure. We have studied the structure of ovule. You know this is the embryo sac, and these are the integuments of the ovule, and inside this what present? Embryo sac present. Inside the embryo sac synergids and XII and antipodal cells are present, and this is a micropylar end, and this is the chalazal end, and this is nucellus, and nucellus which is protected by integuments. Okay, so what is porogamy? Porogamy, you can see here, this one is pollen tube. Okay, the pollen tube which enter into this ovule through micropylar end. Okay, that entry is called porogamy. So porogamy means the entry of pollen tube via micropylar end, through micropylar end of the ovule is called porogamy. Okay, so it is easy to remember. So pollen tube enter through micropylar end, that is called porogamy. And here chalazogamy. Chalazogamy, what it is? Chalazogamy, the pollen tube which enter into this ovule through chalaza. So this part of ovule is called chalaza. So pollen tube enter into this ovule through chalaza, that is called chalazogamy. Okay, the next one, mesogamy. Mesogamy, here you can see these are the integuments of the ovule. The pollen tube which enter into this ovule through integument of the ovule, so that is called, that type of entry is called mesogamy. So there are three different types of entry of the pollen tube into this ovule: porogamy, chalazogamy and mesogamy. Okay, next, entry of pollen tube into this embryo sac. Okay, so how this pollen tube is directed to ovule, ovary, ovule and embryo sac? Because the ovule which produces some chemical substances, that chemical substances are called chemotropic substances. Okay, these chemotropic substances released by ovule which direct the pollen tube towards the ovule. Okay, chemotropic substance. And another structure which is present nearby micropylar end of the ovule, which is called obturator. Obturator, which present nearby micropylar end of the ovule, which also guides this pollen tube towards the micropylar end. Okay, so chemotropic substances and obturator which guides the pollen tube towards the ovule. And this pollen tube which enter into this ovule, which enter into this embryo sac, they make a pore at the tip of their pollen wall, and they discharge the contents present in this pollen tube, that may be vegetative cell and male gametes and cytoplasmic contents, everything into this synergids. Okay, so the contents of pollen tube at the tip of pollen tube, pollen wall, on the pollen wall a pore is formed. Through this pore all the contents present in this pollen tube which goes to synergies. Okay, then what happens, the male gametes are released and cytoplasmic contents are released into this embryo sac. Male gametes fuse with egg cell, another male gametes fuse with polar nuclei, so double fertilization takes place. Next one, double fertilization and triple fusion. Okay, so you already know double fertilization. Double fertilization, which means one of the male gamete which fuse with the egg, another male gamete which fuse with the polar nuclei. So that is called double fertilization. Double fertilization present in angiosperms. Okay, so in 1898, L. Guggenard and S. D. Navashin, they observed in lilium plant and fritillaria that the pollen grains or male gametophyte which releases two male gametes, and these two male gametes, one male gamete which fuse with the egg and form zygote. Okay, that is double fertilization. One male gamete fused with the egg, both are haploids and form zygote, and another male gamete that also haploid which is fused with the polar nuclei formed in the central cell of the embryo sac. Polar nuclei. Okay, polar nuclei which contain two nuclei, and they form primary endosperm. Primary endosperm or PEN. So in this case there are three nucleus they fuse together, they form primary endosperm. So this fusion which is called triple fusion. Triple fusion. So what is triple fusion? Triple fusion, the one of the male gamete which is fused with the polar nuclei and form primary endosperm, that is called triple fusion. Then syngamy, that is one of the male gamete which fused with egg cell and which form zygote, that is called syngamy. Okay, so this all process is called double fertilization. So double fertilization you can observe in angiosperms plants.